Alright, so I have a short announcement regarding the last lecture recording. At the end of the lecture, my software crashed, and unfortunately I was not able to save the recording. And I asked for the recording. One of our students is making the recordings, so thank you, Shant. You can see the link here, so this is the link he provided. So he's willing to share this link with everybody. So if you want to access the recording from the last lecture, you can follow this link. And I've checked this link. You can open it, but first you have to download the file. In order to open it successfully and play the recording, you have to download the file on your laptop. And I've uploaded the instructions how to open this link on the ELC. So there's a Word document with the instructions for this link. So today we're gonna start talking about cell signaling, and we'll focus on the principles of cell signaling. And when we think about signaling, how cells talk, how they communicate with other cells and also with the surrounding environment, those signaling processes can be sometimes very simple, but most of the time they are extremely complex and complicated. And in order to learn about this complexity, we're going to focus first on the principles, on the fundamentals. So we'll define key terms and we'll try to understand the principles before we can understand the complexity of cell signaling. And of course, set of learning objectives and reference to the textbook. So all of this information is in your textbook, chapter 15, and those are the pages. So today we'll talk about key components of the signal transduction pathways. We'll talk about different types of intercellular communication. We'll also talk about distinguish different types of receptors and different types of molecules involved in the communication. We'll talk about positive and negative feedback loops. Those are very important regulatory mechanisms that act on the signal transduction pathways. And I will try to also introduce some important information regarding which will be useful for the next week breakout session. So far, you have a pretty good understanding of cell biology. You are aware that cells come in different shapes, forms, structures, and functions, and they live in different, very diverse environments. From those very simple eukaryotic cells, such as algae or yeast cells, we have a very complex organization of cells in the mammalian human tissues, like this one. This is the lining of our gut. And we've talked about the cells of our gut a lot and their functions. And also neuronal cells are highly specialized cells in our bodies. And we've talked, when we were talking about chromatin and stem cells, we were trying to understand what maintains the cell identity. And we were talking a lot about the different cell-specific gene expression profiles, that those profiles maintain a stable identity of cells. And in addition to gene expression, it's critically important that those cells, every cell, needs to communicate with other cells, with the neighboring cells, and also needs to communicate, needs to sense and respond to the surrounding environment. And this is a fundamental for cell survival. If cells, for some reason, are not able to communicate with the surrounding environment, this is very often a signal for apoptotic cell death, and cell will not be able to survive. And when we talk about cell signaling, very often when you go to the literature, what you see those diagrams, very busy, very complicated diagrams And so this diagram shows a overview of different signal transduction pathways. So this yellow outline is the cell surface, and inside this area is the cell nucleus. And what you can see here, even though you don't know anything, for example, you don't know anything about cell signaling, you see a lot of different. So those are different proteins in the cytoplasm, and a lot of different arrows. And those arrows indicate different interactions between proteins. So when you look at this, the first thing you can think of is, wow, cell signaling is so complex. There is so much action and interaction going on inside the cell, and this is very true. Cell signaling can be a very complicated, very busy process. And today we'll learn how to look at the different cell signaling pathways, and we'll kind of dissect the cell signaling from very basic components and steps. And here, this slide shows the basic steps in the signal transduction pathway. We can distinguish three very basic fundamental steps, which are shown here. So the very first thing that needs to happen in order for the signal to be recognized by the cell has to have a receptor, shown here in green. And this is a specific receptor for specific signaling molecules. So in the extracellular environment, so this could be an extracellular matrix, we have a variety of different signaling molecules, as shown here in red. And the very first step is the binding, the physical interaction of the signaling molecule with the receptor. And when you look at the receptor, it's very important to understand the structure of the receptor. And of course, this is a very schematic representation, but it shows you the fundamental components of the receptor, so we can distinguish kind of three very important domains: extracellular domain, transmembrane domain, and intracellular domain. So you have to remember that for the cell, in order to recognize the signaling molecule, it has to have a receptor that is present on the cell membrane, and the signaling molecule has to directly bind to this extracellular domain. And this is the first step. And very often in cell signaling, when we talk about signaling molecules, another word for signaling molecule is a ligand. So we talk about different ligands, such as proteins, chemicals, gases, and so on. So this is the very important first step, initiating a signal transduction pathway. The second level here is happening in the cytoplasm, when the signal induces a specific changes in the receptor, and those changes in the receptor can be further transduced by other proteins. And this can happen very rapidly, very fast. It's a sequential reaction. Very often it involves not just proteins, but also a broad variety of different small molecules, and those are called secondary messengers. And for example, we'll talk about cyclic AMP, cyclic GMP, PIP2, deacylglycerol, and calcium. Calcium also can be a secondary messenger, playing an important role in transducing the signal from the cell surface to the inside the cell. And here at the bottom, we have so-called effector proteins, which are kind of receiving the signal from those other proteins. And depending on the effector protein, you can have different cellular responses. And for example, the signal can affect metabolic enzyme, and when the effector protein is a metabolic enzyme, we can have an altered metabolism. So the signaling molecule, signal transduction can alter metabolism. Another group of effector protein could be transcription factor, and when this protein is activated, it can lead to a different gene expression or changes in the gene expression. and also cytoskeletal protein, when it's activated, it can lead to change in the shape of the cell, and also it can induce some movement of the cell. So very important 3 steps that you have to remember, and when you study, especially the next lecture, we'll talk about different signal transduction pathways, and when you wanna study those pathways, always go back to the principles and try to kind of like structure your understanding, kind of like, okay, what's gonna happen, what's happening at the cell surface, what is the receptor, and then what are the intracellular components, what is the effector protein, and what it does, how it can affect the cellular response. And we'll talk about a variety of different pathways, but when you have this kind of structure, it will be easier for you to compare one pathway with another, and it should be easier to learn. What kind of changes can be sensed by cells? Virtually any change in the environment can be sensed, and cells can respond to this change in a very specific way. But we can have a general, we can divide those changes into the physical and chemical signals. And the physical signals, cell, for example, can bind to other cell, it can physically interact with other cell or with the extracellular matrix, and this is the example of physical interactions. It can also respond to light or gas, such as nitric oxide. This is a very important regulator in our bodies, and we'll have a separate slide talking about the role of nitric oxide signaling in our cells. And also a variety of chemical signals such as salts, amino acids, neurotransmitters, hormones, growth factors, metabolites, infectious agents, and so on. So anything that you can probably imagine. And it has been estimated that human genome contains more than 1,500 genes for a receptor proteins, for this transmembrane receptor protein. So we can, or our cells can respond to a variety of different signals specifically. Cell communication and cell signaling regulates fundamental processes in a cell. It's critically important for cell proliferation, to regulate gene expression, to regulate different activities such as motility, cell adhesion, cell morphology, and also cell death and differentiation. And for some reason, our normal cells are not able to communicate well with the neighboring cells or with the environment, and something is abnormal about the signal transduction pathways. This can very often lead to a cellular transformation and initiation of the cancer. And variety of different cancers, they have several signal transduction pathways are highly abnormal and dysregulated in cancer cells. So that's what distinguishes cancer cells from normal cells, that they have very abnormal signaling. And now we're going to talk about types of intercellular communications, and we can have two major categories, short range and also long range, but first let's focus on the short range. And one of the examples for the short range intercellular communication is this contact-dependent interactions between two cells. So here in this figure, we can see two cells being in a very close proximity one to another. And this is a signaling cell, this is a target cell. And what you can see, how they communicate, they physically interact. So one cell, the target cell, has the receptor present in the cell membrane. And the signaling cell, it has this signaling molecule shown here in red, but the signaling molecule is not released, it's membrane bound. So there is a direct physical interaction between the membrane-bound signal molecule and the receptor on the target cell. So it's like a handshake between those two cells, and they have to be in very close proximity. And there are plenty of examples for this type contact-dependent cell communication. This is present. Some of the examples are notch signaling and efferents. And those types of, for example, efferents are very important contact-dependent cell communication, which regulates our early stages of embryonic development. Another form of short-range interactions between cells is paracrine interaction, and this is a little bit different from the contact-dependent because here we have a signaling cell that is able to make the signaling molecules and release them to the surrounding environment. And those signaling molecules can act on the receptors of the neighboring cells. And the target cells have to be also in very relatively close proximity to the signaling cell. And there is a form of paracrine signaling called autocrine signaling, as shown here. And what it means, in autocrine signaling, the signaling cell can release this molecule, signaling molecule, and then it will express a receptor for this molecule. So the molecule can act back and can be sensed by the same cell. And this form of signaling would be called autocrine signaling, and this is more talking about autocrine signaling. So we can have a very weak autocrine signal here when we have this single cell secreting the signaling molecule to the environment, and only this cell has a receptor. So only this cell is able to respond to this molecule. And when in this setting the autocrine signal will be very low, versus in this scenario, we have other cells, neighboring cells, are also able to secrete the signaling molecules, and they have also receptors. And in this scenario, we can have a very strong autocrine signal. And as an example for the autocrine signaling, it happens very frequently in the cancer cell mass. Cancer cells are able to secrete epidermal growth factor, and they can kind of support their own proliferation. So this is the example for autocrine signaling. And we also have a long-range interactions between cells. And one of the examples is synaptic interactions. And they happen between the neuronal cells and their target cells. It could be a muscle cell or some other cells. And you may remember that neuronal cells are very specialized cells. They have a so-called cell body and very long extension called axons. And some of the neurons in our body, they can have a very, very long axons, even like a 1 meter long, which is very, very long. And those neurons secrete a signaling molecules called neurotransmitters, and they can be released into this space called synapse. So synapse is the special structure formed between the target cell and the axon of the neuron. So those neurotransmitters are released here, and the receptors are present on the target cell. This could be a muscle cell. So this is a type of long-range synaptic signaling involving neurotransmitters. And another form of long-range intercellular communication is the endocrine signaling. So we have different endocrine cells secreting the signaling molecules, typically hormones. And what happens, hormones can get into the bloodstream, and they can then circulate in the bloodstream, and they can affect different cells in different parts of our body. And this is another form of long-distance interactions. So those target cells will have a receptors for the hormone. And as an example, we can have a insulin hormone produced by pancreatic cells, which can be released, and it gets to the bloodstream, and with the bloodstream it can act on different cells in our body. It can act on liver cells, fat cells, or skeletal muscle cells. And so, we were talking about the principles of cell signaling. And the question for you right now, I know this is an extremely trivial question, but at the same time very important. What is the fundamental component of signal, perception, and transduction? Can you speak louder, please? Receptors, yes, that's great. Transcription factors, why would you say transcription factors? Just the idea. Okay, I will think about it. And you should think about it as well. Okay. Yeah, whatever receptors are the most important components, because they are presented, most of the time, they are presented at the cell surface, and they act like antennas for our cells, and they are able to get the signal and then translate the signal, transduce, initiate the signal transduction. So I like to look at the receptors as like antennas on the surface of our cells. And we can distinguish two major categories of receptors. We can have a cell surface receptors and also intracellular receptors. So we kind of focus on the cell surface receptors, and it has all of those domains. And cell surface receptors are needed for the signaling molecules which have a hydrophilic properties, because hydrophilic molecule won't be able to pass through the hydrophobic membranes, right? So it's critical that it has a specific receptor to bind in order for the signal transduction to happen. But there are also small molecules, hydrophobic molecules, such as some hormones, for example estrogen, and they don't really need the extracellular or cell surface receptor because they are small, hydrophobic, they can easily pass through the membranes, but they still need, in order to have a specific function or effect on the cell, they still need to bind a receptor. And for those hydrophobic molecules, small hydrophobic signaling molecules, we can have intracellular receptors. And they can be present in the cytoplasm, and they can also be transported to the nucleus. Yes, so we have intracellular receptors, and those would be receptors for steroid hormones, retinoic acid, or thyroid hormone. Those are the examples of the signaling molecules that would act on the intracellular receptor. And also nitric oxide receptor has its own receptor that it's located inside the cell. And we'll talk about cell surface receptors. There are different classes of cell surface receptors. We have G-protein coupled receptors, ion channel receptors, receptors with enzymatic activity, and receptors associated with cytosolic tyrosine kinases. So we'll start talking more in detail about G-protein coupled receptors today, and then the next lecture will cover all those additional classes of receptors. And here I wanted to talk a little bit about this very interesting superfamily of steroid intracellular receptors. So you can see on this panel a variety of different receptors. A receptor for the cortisol, for estrogen, progesterone, vitamin D, thyroid hormone receptor, retinoic acid receptor, and this is the structure of those receptors. So those are short polypeptides, and they have a specific specific domains. Maybe I shouldn't say polypeptides because they are probably like bigger proteins. But what they have in common, they have this DNA binding domain. And actually we can distinguish three modular parts in those receptors. They have a hormone or ligand binding domain, they have DNA binding domain, and they also have a transcription activating domain, or yes, this is the transcription activating domain. And this is the structure of this receptor. So here we have in green the DNA binding domain, the N-terminal transactivation domain, and here there is a ligand binding domain, and you can see it has two parts, this light blue and darker blue. And in this conformation, this ligand binding domain doesn't bind any ligand, and it's typically associated with the inhibitory protein, which keeps this conformation of the ligand binding domain kind of like open. And in this form, the receptor is inactive, but when there is a ligand molecule that can be bound specifically by this receptor, it can bind here within those domains. So those domains will change the conformation, kind of like surround the signaling molecule, surround the ligand and bind it inside. And this entire protein and this entire receptor will change its conformation. Now, the DNA binding domain will be able to interact directly with the specific DNA sequence. So this entire receptor now can bind the DNA, and of course its transactivation domain can be active and it can interact with other proteins, and this can lead to changes in the transcription of the target genes. So this is conformation of the active receptor. And this is just the example showing a different activation of a different receptor, the thyroid receptor. So it's sitting here on the DNA in its inactive form, just waiting for the thyroid hormone to bind. So in this form, the specific sets of genes are regulated by the thyroid hormone receptor are not expressed. And when the hormone is available, it binds, activates the receptor, and it can lead to a transcription activation. And now I want to talk about the interactions between the signaling molecules or ligands and receptors, because this is very important first step. Like I said, it initiates entire signal transduction pathway, and it's very important to understand how this interaction happens. So this is, in here there is a growth hormone shown here in red, and this is a receptor for the growth hormone. And so the interactions are very specific. So growth hormone is able to bind specific amino acid residues on the receptor, and the binding involves a weak bonds such as hydrophobic, ionic, or van der Waals, but there can be a variety of those bonds. So it's not just single bond. It can be a maybe like 100 different weak bonds, and this all contributes to the strength of the interactions and also the specificity of the interactions between the ligand and the receptor. And it has been determined, for example, that 2 tryptophan residues in the receptor, so just two residues in the receptor, are responsible for most of the energy to bind the hormone. But of course other amino acids are also important and are involved. But what researchers found that 2 amino acid residues are critical and very important for the effective and strong binding of the growth hormone to the growth hormone receptor. And when we talk about interactions, ligand-receptor binding, we very often talk about affinity of interactions. Affinity, in other words, means strength of the interactions, And this can be described in those very simple formulas. So R stands for receptor, L is ligand, and the K on, K off, those are the association and dissociation constants. So this K on is the rate of the formation of ligand-receptor complex. So this is association, and this reaction backwards means a dissociation. So dissociation constant, Kd, is a standard measure of the affinity. And we can distinguish we can have a high affinity receptors and low affinity receptors. And by looking at the dissociation constant, we can kind of determine what kind of receptor it is. Does it have a high affinity or low affinity for the ligand? And when you look at this formula, when we have a high dissociation constant, it typically shows that means that the interactions between the receptor and the ligand are weak, because there is a very high dissociation of the receptor and the ligand. When this value is low, that means that the affinity of the receptor and the ligand is very high, because they will form the complex and the ligand won't be released so easily from the complex. And depending on the affinity of the receptor, we can have a molecule such as hormones, and the concentration of the hormones in our blood can be very small, and they are circulating in the blood, and because the receptors for hormones, they have very high affinity for hormones, we can have a very effective cellular response when cells are exposed to hormones, even though the hormones are at very low concentration, but because of and they have to travel long distances, but most of the time the receptor for hormone has a high affinity, so it can bind in a very strong the hormone ligands. Therefore, we can have a very, very strong and specific responses. And here I wanted to talk a little bit about the competition assay or ligand displacement assay. This will help you in the next breakout. So this is how we can look at the interactions between the ligand and the receptor. So this schematic shows a cell, and this yellow thing is the receptor presented on the cell surface, on the cell membrane, and the green is specific ligand, and in this case radio labeled. So it binds specifically. There is a strong interactions or specific interactions between the radio labeled ligand and the receptor. And in this assay, we can test different drugs and different molecules. Those are called competitors. So they will compete with this native ligand, the radio-labeled ligand. They will compete for binding to this receptor. And then we can evaluate different chemicals, different drugs for their ability to bind to the receptor. And so what we are looking here at, we have the cell that has the radio-labeled ligand, and we add those competitors, and we incubate the cell in the presence of those competitors, and wait, and measure the level of this radio-labeled ligand. So over time, some of those competitors will be able to displace the native ligand and will bind here. And the radioactivity, the level of the signal will drop. We can monitor this. So this is a typical graph that you would get in this type of assay, competition ligand displacement assay. So on the y-axis, we have a activity of this radio-labeled ligand, and on the x-axis we have a increasing concentration of the competitor, And what's going to happen over time when you increase the concentration of the competitor, it's progressively replacing the ligand, radiolabeled ligand, so the radioactivity drops. And this is a competition curve. And very important metrics here is the IC50. And the IC50 means the concentration of the drug able to displace 50% of the native ligand. So this is a very important metric. And when we look at different drugs, we can compare their affinity. So when you do the experiment, you would see a different curves. And based on these curves, looking at the IC50, you can kind of determine the strength of those competitors and the affinity. So which has the strongest affinity to the receptor, which has the lowest. And we'll talk about that during the breakout next week, but I thought this could be very useful information for you to have. And cells can respond to different signals in very specific ways. So typically, a cell is exposed to 100 different signals, and of course is not able to respond to every signal. It's very selective, and of course we've talked about the receptors, so it has a specific receptors, right? And this figure shows you a very different responses depending on the types of the signals that cell is exposed to. And here in this first scenario, cell is exposed to signals A, B, and C. And this could be a very good, very nice environmental signals and showing that there is everything that cell needs and it can survive. So this could just mean to the cell, okay, I am happy here, this is a good environment, I can survive. In this scenario, we have those signals, A, B, and C, which could be just the water, nutrients, and oxygen, because those are the essential signals in the environment that would be needed for the survival. And here we have additional signals, D and E. Those could be growth factors. And the cell, it would act on the cell and tell the cell that it's time to grow and divide, because the environment is so good, so nice, we can grow and divide. And in the absence of those D and E, but in the presence of new signals, cell can make a decision about the differentiation. Because, for example, environment is changing and the specific function of the cell would be needed, so cell can differentiate. In case, in the absence of all of those essential signals, cell can undergo programmed cell death, because the environment is not good, cell won't be able to survive, and it can activate a programmed cell death called apoptosis. So there are different responses to those different signals. And this is a very nice example showing how a single molecule, a neurotransmitter called acetylcholine, shown here, is a neurotransmitter secreted by the neuronal cell, how this neurotransmitter can have totally different effects on different cells. So it can act on different kinds of cells. We can have a heart cells, salivary gland cell, or skeletal muscle cells. And all of those cells, those are different cells, right? And they have the same receptor. So we have the same molecule, the same receptor, and totally different responses. So when it acts on the heart, it can lead to a decreased heart rate. When it acts on the saliva gland cell, it can lead to secretion of saliva. And on the muscle, it can lead to contraction. This is very important. When we talk about cell signaling, we often use those terms, agonists and antagonists. And make sure you understand those terms and know the difference. So agonists, what are the agonists? Typically, agonist is a drug, and it's a kind of like a synthetic analog of the natural ligand. So what this synthetic analog can do, it can bind the receptor and it can activate, like fully activate them. And typically what agonists do, they mimic the function of the natural ligand. So they are very similar in structure to the natural ligand, but they are not the natural ligand. They are typically drugs or some synthetic compounds. Antagonists are drugs that can bind the receptor but do not activate the receptor, and instead they block the receptor, so they inhibit the function of the natural ligand. And this figure shows you the differences and different responses when you look at the agonist and antagonist. So here we have an agonist acting alone, binding the receptor and leading to the full activation of the response. In this scenario, we have antagonist that is able to bind and it blocks the receptor, so its natural ligand won't be able to bind, and this leads to a inhibition or no activation. And in some cases, we can have a combination of agonists and antagonists, and this combination would produce somewhat intermediate response. And now I want to kind of introduce the different categories of cell surface receptor proteins. So there are three major categories. Class 1 are ion channel coupled receptors, shown here in the orange. So they are very simple membrane receptors, and they can be regulated. So the activity of this receptor can be regulated by signaling molecules, and often, for example, neurotransmitters such as acetylcholine can bind here the receptor and can activate the receptor. So they form the channels in the cell membrane and will facilitate import of different ions. And we're not going to talk much about this class, but I wanted to briefly mention about this class. And this class of receptors are involved in the very rapid synaptic signaling between nerve and muscle cells. And yeah, like I said, are mediated by neurotransmitters. Class 2 are G protein coupled receptors, and this is a very abundant class of receptors in our cells, and we'll focus majority of our attention during today and the next week. We'll talk about this class of receptors. So they are very different from the class 1. They have three major components in the plasma membrane, and they operate through interactions between those three different components. So we have a receptor shown here in green. We have an inactive protein called G protein and inactive enzyme. And when the signal molecule comes, it's changing the conformation of the receptor, and also it leads to activation of the G protein, and then the signal can be transduced to this effector protein. So we have some additional interactions going on. And we'll talk about details of this process a lot. And another class is enzyme coupled receptors. So they are present as a monomers, and they have the specific thing that distinguishes this class from the previous class is that they have a inactive catalytic domain in the cellular, in this intracellular region, and they are activated by the signaling molecules, and kind of like two monomers are brought together, and this close proximity of the monomers together leads to a transactivation of the catalytic domains. And then, so this is the way those receptors will transduce the signal. So they will activate this domain, and then this domain will be able to activate some downstream proteins. And I wanted to talk about so far we were talking about the receptors and what's happening at the cell surface. And now we're going to just move on and focus on what's happening downstream. And there are proteins called molecular switches, and those are very important proteins that are involved in the signal transduction. So they sit inside the cells in the cytoplasm. So they can be associated with the cell membrane, but they do not have like extracellular domains like receptors. And I really want you to encourage to study those, the mechanism, and you have to understand how molecular switches work. There will be a question on the exam concerning those, the molecular switches. So we have two categories of molecular switches, and those are the proteins that can switch from from the active conformation to the inactive conformation, and they are involved in the signal transduction. And one category of protein is regulated by phosphorylation. So when the signal comes, here we have this inactive molecular switch, and the signal will lead to activation of protein kinase, and protein kinase will phosphorylate the switch. And this phosphorylation will activate the switch, and then the signal can be transduced to some other proteins. And when this is active, it can be deactivated by protein phosphatase. So the key thing about molecular switches is that they they switch from active to inactive form all the time, and they are regulated by specific proteins that can activate and inactivate them. So phosphorylation is one way to regulate the molecular switches, and another way is through GDP and GTP binding. So when in this conformation this protein is not active, GDP is bound, and then when the signal comes, there is a exchange of the GDP to GTP, and now we have a activation of the protein, and then signal can be transduced into the downstream targets, and this molecular switch has to circle back to the inactive conformation. Let's see. And we have a slow and rapid responses to the extracellular signals, and this diagram shows the distinction between the slow and rapid. So this pathway is very fast and rapid. It can happen in seconds to minutes. And this pathway, shown here by green arrows, it's slower. It can happen in minutes to hours. So when you look at this diagram, what's the main difference between this red arrow pathway and the green arrow pathway, and what it means? Why this one is fast and why this one is slow? What is this structure here? Yes, it's the nucleus. So this pathway is not going through the nucleus. And what's happening in the nucleus? Yes, so we have transcription. So in this pathway, so why this pathway is slower? Exactly, that's correct. Transcription takes time. So in this pathway is slower because it doesn't have all of the key components just, ready in the cytoplasm and waiting for the signal. So it has to make the downstream components. Therefore, it's slower. It's going through the nucleus. It requires some changes in the gene expression. It needs to make some proteins. Therefore, it takes time and it's slower. And these pathways can happen very fast because all of the components, the downstream components, are present in the cytoplasm, and it just happens like a very fast, like a sequential reaction. Everything is ready, and it can happen in seconds to minutes. So this is a major distinction when we talk about slow and rapid responses. And cells can produce a different rates of responses. And this diagram shows the different types of responses in different cells. And here the y-axis is the response rate from the slow to fast. And here on the x we can see the concentration of the signal molecule from low to high. And those different curves represent different responses. So in the hyperbolic type of response, what's happening? We have a increasing concentration of the signaling molecule, and the cell, when it has more signaling molecule, the response is higher, but it's kind of like proportional to the signaling molecule. The sigmoidal type of response is that at the beginning, the response is very slow, especially when the concentration of signaling molecule is slow. Then we have a window of time that during this window of time, the response is rapidly, it's much faster at the specific concentration or range of concentrations, and then it kind of plateaus. And this is a very interesting response. It's called all or none, shown here in green. And what it is here, you have a very low concentration of the signaling molecule, nothing is happening. And then at specific concentration, it's still low, but it's very specific, so it's reaching the threshold, and then we have a full activation of the response here at this specific concentration. And when we increase the concentration later, the more molecules are, they don't gonna affect the response because the full response is happening here very rapidly. And when we talk about signaling, we talk about positive and negative feedback regulatory loops. And those signal transduction pathways are regulated by those loops, and you have to be able to distinguish and kind of define what is the positive, what is the negative feedback. So in the positive feedback, the thing is that the output of the process can act on the previous steps, and it can lead to a stimulation of its own production. In the negative feedback, the output acts back on the previous steps, and it inhibits its own production. And this is kind of like demonstrate the positive and negative feedback. It's in your textbook. We don't have much time today to discuss this, but you can. So this is kind of how it works when we look at the activation and inactivation of the specific enzyme. And here I wanted to talk about the very important process in the cell signaling. So for example, when we have a cell and the cell is exposed to a specific signal for a very long time, what's going to happen? The cell will decrease its response to the signal. So a very good example is that, for example, when we are walking outside in the bright sunlight, and we are walking for like maybe an hour or so, and then we enter the dark room, like this classroom. So for a few seconds, what we see, we kind of see that it's much darker in here. And what's happening when we were walking outside, our photoreceptors in our eyes were deactivated because those receptors were exposed to the bright sunlight for a long time, and they lost their sensitivity. So when we get inside this room, it's gonna take a few seconds for those receptors to adjust to the new lighting. And at the molecular level, this is a very important process. It protects cells from overreacting to signals, and also ensures that cells are able to detect and respond to changes in the environment, not necessarily detect and respond to specific strength of the signal, but what's important is the change. So cell has to be able to detect changes in the environment. And at the molecular level, we can have a different ways leading to a desensitization of the receptor. So when we'll desensitize the receptor, this can stop the response, even in the presence of the signaling molecule. And all of those panels here show different ways of receptor desensitization. So in one way, we can have a receptor sequestration, where a receptor and the binding molecule here, the signaling molecule, will be sequestrated, will be kind of like, will form the vesicle and will leave the cell membrane, will be in the cytoplasm. We can also have a receptor downregulation, so it will be like completely degraded in the lysosome. And we can have different forms of inhibition or receptor inactivation. So there are other proteins inside the cell that can bind either to the receptor or to the downstream steps, and it can inhibit specific steps in the signal transduction pathway. And again, this is a very important process, and there will be some questions concerning this process on the exam. And the underlying mechanism for this process is a negative feedback. We probably gonna skip the activity because I have some other slides. So now I'm gonna start talking about G-protein coupled receptors, and this is a new set of learning objectives, and I will supplement those learning objectives on the next lecture. So this right now, we're going to focus on this category of proteins. And let's see, I just want to see how many slides I have. Quite a few. Okay, so G-protein coupled receptors. Very important thing. So this is the receptor. This is the structure of the G-protein coupled receptor. And what it is, it's a protein, a very long protein, single protein, and it's here embedded in the cell membrane. It has the N-terminal domain, and very specific thing here, it has a 7 transmembrane domains, and it has some additional domains that are sitting in the cytosol. And important thing to remember is that G-protein coupled receptors are very conserved, and they have, all of those receptors have 7 transmembrane domains, all of them. So not 3, not 11, they have 7 transmembrane domains. And we focus on those receptors because they are the largest family of the cell surface receptors. They have more than 800 G-protein coupled receptors. And yeah, like I said, this is a single polypeptide with 7 transmembrane domains. So this receptor doesn't have any intrinsic enzymatic activity, but it can interact with other proteins that have. And there is also feedback mechanism associated with the desensitization of this receptor. We'll talk more about that in the next lecture. And G-protein coupled receptors are present in various tissues of our bodies. They are highly abundant in our brain, in our eyes. We can see because of G-protein coupled receptors. We can smell. The sense of smell is mediated by G-protein coupled receptors, and our immune system relies on the G-protein coupled receptors. And we'll talk about vision and smell, how those receptors mediate all of those senses. They regulate behavior and mood. They are very abundant in the brain, and of course they regulate the immune system. And they have some new roles, like they can regulate cell density. This is a novel role for G protein couple receptors, and also a water balance or homeostasis in our body. And 50% of all known drugs right now available on the market act on G protein couple receptors, on different aspects of signaling through G protein couple receptors. And this is why this is so important to understand. And this figure shows a phylogenetic tree of all of the 800 G protein couple receptors of the human. And this phylogenetic tree was created by a collaboration between different labs. And people want to understand as much as possible because we can discover novel drug targets, and we can, by knowing the structure and function of those receptors, we can open new doors for novel treatments and novel drugs. So it's important. So G protein is a trimeric GTP binding protein. So it's not a receptor, but it's a very important protein that interacts with the receptor. And you have to know the structure of the G protein. So you have to be able to describe what is the structure and how activation of the receptor changes or affects the structure. So we have three subunits, alpha, beta, and gamma, and alpha subunit. And in this form, G protein is inactive because it binds the GDP. And G protein is the perfect example of molecular switch. So when we are talking about molecular switches, this is the example of molecular switch. And in this form, it's inactive, it binds GDP. And there are various types of G proteins, very specific for the particular receptors. And we'll talk about those different proteins. And not all of them, only those that are highlighted in the red frames. And so this is also very important information concerning the breakout session. So we have a G protein couple receptors that are responsive to a stimulatory hormones such as epinephrine, and those are called adrenergic receptors. In this figure, they are present on the same cell, but in the real life, they are typically present on different cells. And what's very interesting about those receptors, they interact with different types of G proteins. So one type of receptor can interact with the stimulatory G protein, and these interactions will lead to a activation of this enzyme, adenylyl cyclase. And different receptor can interact with so-called inhibitory G proteins, and when this happens, this leads to inactivation of the adenylyl cyclase. And we'll talk about this process the next lecture. I'm going to skip the video. And here I wanted to show you, this is the last slide, and then we'll have a short quiz. So here I wanted to show you this because this is important for the next breakout. So this is a different view of the adrenergic receptors and activation of the adenylyl cyclase. And this system is important. So when we are trying to understand the interactions between the receptors and the G proteins, and the question we are asking, okay, how do we know which part of the G protein couple receptor interacts with G protein? So we know the structure of those receptors, right? 7 transmembrane domains. And so this blue one is for this adrenergic receptor that is leading to inhibition of the pKa, or the adenylyl cyclase. Oh, the pKa is the downstream enzyme here. So this is the adenylyl cyclase. And second receptor leads to activation. When it interacts with the G protein, it leads to activation. So what researchers can do, they can take advantage of the system, and they would be doing experiments in frog oocytes. So they have the ability to create so-called chimeras when they combine some domains from one type of receptor and other domains from a different type of receptor. So they can kind of address this question by creating those different chimeric receptors and looking at monitoring the activity of the downstream enzyme, in this case PKA. So when they kind of change the domains, here we have 1, 2, 3, and 4 domains are from this from the one receptor, and 5, 6, 7 are from the different receptor. And then when we look at this receptor and the activation of the PKA, we can kind of like conclude, comparing different chimeras, which domain, which part of this entire receptor is able to directly interact with the G protein by comparing and analyzing those different chimeras. And we'll talk more about that, about the experimental design and how we do these experiments during the breakout session, because we don't have time really to talk about that. But the conclusion for this experiment, and when you will have some time at home, you can analyze this. So the conclusion for this experiment is that domain 5 is the domain that directly interacts with G protein. Okay, so I'm gonna stop here and we'll have a short quiz right now.